Greetings everyone, and thank you for attending our presentation. Today we will be discussing our recent work on absorption driven drug formulation.
The goal of formulation development is to create a complex drug delivery system for the API that meets numerous criteria. These criteria includes manufacturing, stability, compatibility, safety, economical, legal, or performance-related metrics. In order to be successful, a formulation and a drug product must meet all of these criteria. During the formulation process, multiple excipients and formulation strategies are tested, selected, and optimized based on the predictions for the various metrics. In vivo performance is one of the most challenging parameters to predict and control during the process, and one of the last criteria to be tested in the form of bioequivalent study or other in vivo human clinical trials. A method for rapidly predicting in vivo performance continuously during the development process is highly desired, as all changes to the composition or the structure can affect the performance. So any alteration to the formulation for manufacturability, stability, or other reasons potentially invalidate former predictions. Our research for the past few years has been focused on understanding the absorption process and the physicochemical parameters that can characterize it. Furthermore, we aim to develop in vitro tools and applications to measure these parameters in order to improve the quality of the predictions. Since most of the active pharmaceutical ingredients are absorbed by passive diffusion, in this presentation and also in our work, we focus on modeling simultaneous dissolution and absorption by passive diffusion. As a starting point, let's take a look at the in vivo process after drug intake. The active pharmaceutical ingredient first has to dissolve from its matrix, as only the API in solution is able to cross biological membranes. Upon transport through several lipophilic barriers of the gastrointestinal tract, the API reaches the bloodstream. This is called absorption. As the API is traveling in the gastrointestinal tract, various amounts is getting absorbed as a function of flux through that specific membrane, transit time, and available surface area as primary parameters. In this presentation, we are focused on modeling the flux parameter and do not consider any potential formulation effect on transit time or available membrane surface. As we focus on flux, let's see what we mean by that. Flux is a measure of transport and describes the amount of API that has passed through the membrane. By definition, it is the net number of moles of particles crossing unit area per unit time as seen in the equation above. The mass change can be expressed as concentration change over time multiplied by the volume. To visualize the meaning of the equation, we have plotted a concentration time curve depicting the concentration change at the apical side. It shows that flux is directly proportional to the slope of the curve. Circling back to the bigger picture, dissolution and absorption are consecutive processes in vivo. Recently, a number of poorly water-soluble drugs is increasing. Therefore, novel formulation strategies aim to improve the dissolution process. But whenever dissolution is altered, it has an effect on the absorption as well.
This phenomenon is often referred as the solubility-permeability interplay. In the following slide, I would like to sum up the great work of the group of Eric Dahan and Jonathan Miller. They found in vitro and in vivo evidence as well that whenever solubilizing agents like surfactants, lipids, cyclodextrins, or co-solvents are used, the solubility of the API increases, but at the same time its permeability decreases. For that reason, whenever we aim to improve the bioavailability of the compound, the trade-off between solubility and permeability has to be considered. When using the amorphous form of the API, the higher concentration in solution is achieved via supersaturation. Because this way the thermodynamic solubility of the compound is not altered, the permeability stays also unchanged. This may be the true advantage of amorphous pharmaceuticals from bioavailability point of view. To gain a deeper understanding of solubility-permeability interplay, a schematic figure of the permeation cell is shown on this slide. The donor and the acceptor compartments contain aqueous media and are separated by lipid membrane. The API first dissolves in the donor compartment, then it must partition into the lipophilic membrane, where it diffuses, then partitions again into the acceptor phase. If we describe these processes with partitioning coefficients and fix first law, and assume that the solubility in the donor and the acceptor media is constant, then flux can be described as the product of permeability multiplied by the concentration difference between the donor and the acceptor phase. That form of the equation shows that the driving force of membrane transport is the concentration gradient. The permeability is a constant which describes the membrane transport of the API. If we take a look at the experimental data of various meloxicam formulations on the right-hand side graph, showing flux versus donor concentration, then we can see that flux is truly directly proportional to the donor concentration for every meloxicam containing formulation individually, but the slopes in the diagram differ, meaning that permeabilities differ as well. Once again, this data shows that using different formulation additives might change not only the solubility but also the permeability of the compound. And more importantly, it points out that permeability is not a constant parameter of the API in this case. This means that the driving force of membrane transport cannot be simplified to the concentration gradient in cases where different solubilizing agents are used. The reason for this is that we made the assumption that the solubility in the donor media is constant, but this is not true when solubilizing additives are used. We may look at the same phenomenon with a different physicochemical model. In this slide, we show a physical-chemical description where we do not assume that solubilities are constant. The result shows that the driving force of membrane transport cannot be simplified to the concentration gradient. The difference in degree of supersaturation, defined as the ratio of donor concentration of the drug to its thermodynamic solubility between the donor and the acceptor side, was found to be the real driving force of membrane transport. This description also explains why the use of formulations that create supersaturated solution is desirable. Amorphous solid dispersions might be advantageous from bioavailability point of view because they increase the driving force of membrane transport.
As shown in the example of meloxicam, flux assays, or dissolution permeation assays, are suitable to investigate the solubility-permeability interplay and any related formulation effect, as they allow us to simultaneously characterize the concentration change in the donor side over time and the transport through the separating membrane. In this presentation, our main objective is to introduce a concept called Absorption Driven Drug Formulation that applies flux assays in all stages of formulation development to gain a better control on the flux provided by our formulation, allowing us to more accurately predict the in vivo performance of the developed formulation. The ADDF concept utilizes three types of flux assays throughout the development process. Initially, 96-well plate-based flux assays are done to characterize the possible interaction between the API and the excipient. In the next stages, small volume flux assays are done to understand the behavior of more complex lab formulations or pre-formulation samples and to compare various formulation strategies. In the final stage of the development, biorelevant volumes are used in the donor side to investigate and compare final dosage forms. The initial 96-well plate-based flux assay investigates API and excipient mixtures to study their interactions. These simple assays are run in biorelevant media and compare the flux of the pure API to the flux when the excipient is also present in the solution while keeping all other parameters unchanged. The test can have three outcomes. The first case is when the API has the same flux with and without the excipient. In this case, the excipient has no effect on solubility and permeability of the API, or the gain or loss in solubility is compensated by the opposite effect in permeability. The second case is when the flux is higher without the excipient. In this case, the solubility loss is not compensated by the permeability, or the permeability loss is not compensated by solubility gain. The third case is when the flux is higher in the presence of the excipient. Consequently, the solubility or permeability gain is not or not completely diminished by the decrease of the other parameter. The generated data can be used to select the initial excipient to be used in the formulation, or later on, as changes are required, the impact can be better predicted, minimized, or compensated for. In the next stage, small volume flux assays are done to characterize more complex samples in later stages of development. The assay is carried out in 16 to 20 ml donor compartment volumes, and biorelevant media is used to mimic either fasted or fed state conditions. Due to the flux cell arrangement and volume conditions, continuous concentration monitoring can be applied in both compartments, providing much more insight into the dissolution and permeation process. Various solid or liquid samples like physical mixtures, granules, pellets, or solid dispersions can be studied, and the effect of individual excipient or excipient concentration changes or formulation process alterations can be investigated. The performance of the samples can be compared by calculating flux based on the slope of the appearance curve in the acceptor chamber, or by calculating the total permeated amount based on the area under the curve. In the final stage of formulation development, finished dosage forms like capsules or tablets are compared. The large volume flux assay can be configured with the donor volume of 250 or 900 ml and therefore is suitable to investigate the whole dosage form, meaning it can provide information on the tablet or capsule disintegration as well as dissolution, solubility, and flux-related parameters. In the late stage of development, usually only small modifications are applied to the formulation, so the biorelevant assay conditions, especially donor media and media volumes, has an even bigger importance. As in the small volume setup, real-time concentration monitoring in donor and receiver compartments can help to characterize the processes. If the system is used for generic formulation development, then in this stage, the brand formulation can be investigated in the same assay setup and used as reference in order to make more accurate predictions for the in vivo situation. The overall concept is that incorporating flux measurements in the early and late stages of the formulation process allows scientists to have better control of the flux of the API in the final dosage form and ultimately to be able to predict in vivo study outcome. For the next part of the presentation, we will share case studies on each flux assay setup and will show a simple example of the generic formulation development based on ADTF concept. The first set of case studies will focus on API-excipient interactions studied on Pampa platform. Back in the early 2000s, Pion and Hoffman-LaRoche were collaborating to develop a 96-well plate-based technique to predict API-excipient interactions. The first approach involved two separate assays, a plate-based solubility assay called Microsol and the Pampa assay. In both assays, the solubility and the permeability were measured with and without the excipient, and differences were evaluated at 3 donor pHs in the range of 5 to 7.4. The solubility and permeability data were combined based on the flux equation, and the flux profile change was evaluated. As the example of clotrimazole shows, 1% hydroxypropyl beta-cyclodextrin had a slight negative effect on permeability and a significant positive effect on solubility, so flux enhancement can be expected if this excipient is applied in a 1% concentration range. The concept appeared to be suitable for the evaluation of the trade-offs between solubility and permeability, but it required a substantial amount of work to measure both solubility and permeability with and without the excipient, and in case of low-soluble compounds, it was particularly challenging to get accurate solubility data. Therefore, the approach has been adjusted and the solubility and permeability assays were combined into a single plate-based assay on the Pampa platform. In this assay, the solid APIs are placed in the bottom compartment of the Pampa plate. Donor media, with and without the excipient, is then added to the solid API immediately before placing the top plate, which contains the GIT lipid and the acceptor phase, onto the bottom plate. After the selected incubation time, the plates are separated and the API is quantified in the receiver compartment only. Whether the presence of excipient in the solution improves or impedes the absorption properties of compounds, or does not change them at all, is quantified by comparing permeation as measured by the receiver UV absorption. These examples show the UV absorbance as a function of wavelength for various tamoxifen samples, where the effect of 5 excipient have been studied and one of them has been run in 2 different concentration levels. The top left corner shows tamoxifen permeation without excipients. Lower UV signals mean a negative excipient effect, same signal intensity means zero gross effect, and higher intensity signal means positive effect. On the example, 3 millimolar sodium taurocholate and 1% hypromellose has a negative effect, 1% propylene glycol and 1% N-methylpyrrolidone has zero gross effect, and 50 millimolar sodium taurocolate and 1% HPBCD has a strong positive effect. It is interesting to note that for sodium taurocolate, 3 millimolar concentration decreased the flux, while 15 millimolar had a 4 times increase compared to the pure API flux. This experience reinforced that excipients can have a concentration-dependent effect on API flux, and studying excipient effect at multiple concentration levels is recommended. Avita Baig and co-workers have published the next case study in 2015, where they have conducted a head-to-head comparison of different solubility-enabling formulations on the PEMPA platform. Etoposide was formulated in several strengths of 4 solubility-enabling formulations: hydroxypropyl beta-cyclodextrin, the cosolvent propylene glycol 400, the surfactant sodium lauryl sulfate, and an amorphous solid dispersion formulation. In the PEMPA measurements shown on the left-hand side, they have found a decreasing permeability with the increased amount of HPBCD, PEG 400, and sodium lauryl sulfate. The right-hand side graph demonstrates that the solubility increasing effect of this excipient has been compensated by the permeability decrease. The amorphous solid dispersion samplers also provide a significant donor concentration increase, but the permeability remained the same despite of the increasing load of the solid dispersion and therefore increasing amount of copovidone as formulation excipient. If you look at the theoretical flux as a function of solubility or maximal donor concentration, the amorphous solid dispersion provides an outstanding advantage over the other formulations, as the increase in the dissolved amount actually provides a proportional increase in the flux as well.
In the following chapter of the presentation, I'm going to talk about case studies carried out on small-scale dissolution permeation apparatuses. The first example is the ranking of hydroconazole containing formulations. The aim of this study was to rank order various enabling formulations of HYDRA with the help of small volume flux apparatus and compare the results to in vivo pharmacokinetic data. For that reason, formulations containing the API in a crystalline form, such as micronized powder and nanosuspension, and also soluplast containing amorphous solid dispersions were created and tested against the commercially available amorphous solid dispersion of HYDRA, the so-called Sporanox capsule, and the untreated API. The above-mentioned formulations were tested with microflux apparatus. HYDRA capsule is supposed to be taken after a meal, therefore the donor compartment was filled with fessive media, while the acceptor representing the bloodstream was a pH 7.4 solution. The separating membrane was painted with GIT lipid mixture. The concentration of the API in the aqueous compartments were monitored with UV probes. The concentration time curve of the acceptor chamber shows that the flux of the amorphous formulations of HYDRA, soluplast containing and Sporanox, was found to be significantly higher than any of the crystalline forms. The Sporanox formulation showed the highest initial flux, but after 2.5 hours it was reduced to a similar level to the nanosuspension formulation. Although initial flux of the soluplast formulation was lower than for the Sporanox, it was not reduced as drastically as the latter after 2.5 hours. The total amount of HYDRA in the acceptor compartment after 240 minutes is shown in the left figure. Although the permeation kinetics of the two amorphous formulations are very different, from the accumulated mass point of view, they are almost the same. The accumulated mass in the acceptor chamber was correlated with in vivo rodent AUC results. They showed the same rank order as the rodent AUCs of these formulations reported in literature. This work demonstrated that in vitro flux measurements using lipophilic artificial membranes could correctly reproduce the rank order of PK results for HYDRA formulations.
Let's see another example of the use of small volume flux assay in formulation development. In many cases, one formulation technique is not enough to create an enabling formulation of poorly water-soluble drugs. Therefore, the aim of this work was to investigate the in vitro dissolution permeation properties of a complex formulation matrix using flux assays and evaluate the effect of different formulation techniques on flux. Various formulation strategies were combined to create amorphous solid dispersions using electrospinning to get thin fiber structure, sulfobutylator beta-cyclohexene as a solubilizing agent, and citric acid as a microenvironmental pH-modifying agent to enhance the dissolution and the bioavailability of a poorly soluble antipsychotic drug, aripiprazole. The aripiprazole containing amorphous solid dispersion and its physical mixture were compared to the neat API by a simultaneous dissolution permeation assay. The concentration time curve of the donor chamber shows that by physically mixing the formulation additives like citric acid and sulfobutylated cyclohexene significantly enhances the dissolution of the API, the amorphous solid dispersion formulation shows even greater enhancement and creates an ultra-fast drug delivery system. Even though such high supersaturation is not stable, it produces enormous improvement in the flux across the membrane, as can be seen from the concentration in the acceptor chamber. Approximately 50 times more molecules went through the membrane from the electrospin formulation matrix than from the crystalline form. With the help of the physical mixture, it is possible to visualize the contribution of formulation additives and amorphization separately. In conclusion, in case of combining different formulation techniques, the solubility permeability interplay has to be considered, and a small volume flux assay might be of a great value when evaluating the effect of each technique separately.
In the following chapter, I'm going to present case studies carried out on large-scale dissolution permeation apparatuses assaying final dosage forms. Let's see another Hydroconazole case study, but this time Hydroconazole, a BCS2 classified antifungal drug, was chosen as the model compound for the prediction of food effect and bioequivalence from final dosage forms. The first commercial solid formulation of Hydra, Sporanox capsule, contains the API in amorphous dispersed with hydroxypropionmethylcellulose non-sugar spheres. This formulation uses the advantage of high surface due to the small spray-dried particles dispersed on the sugar spheres, and also the amorphous state to improve drug dissolution. The capsule formulation is supposed to be taken after a full meal, which limits its utility as patients being unable to take solid food are excluded. In contrast, Sporanox oral solution, marketed 5 years after the capsule, has shown higher bioavailability in fasted state than in fat state, therefore may be advantageous in therapy. The oral solution contains 10 mg per milliliter Hydra in a pH 2 media with an extremely large amount, 400 mg per milliliter of hydroxypropyl beta-cyclodextrin. The latest marketed formulation of Hydra is Hydroconazole, or also called Lozanok, which is a novel amorphous solid dispersion containing Hydra in a pH-dependent polymeric matrix, hydroxypropionmethylcellulose phthalate, to enhance its dissolution and intestinal absorption. Although with halved a dose compared to the innovator product Sporanox capsule, it has failed to meet bioequivalence criteria, but showed therapeutic equivalence. The manufacturer of Hydra found no significant food effect during in vivo studies, therefore this formulation can be taken before or after meal as well. The first step of the project was to carry out flux assays of the 3 Hydra-containing formulation in fasted and fat state with the so-called bioflux apparatus. In this device, a receiver chamber with integrated permeation membrane, overhead stirrer, and fiber optic UV probe was inserted in the 500 ml vessel of the USP2 apparatus. The working volume of the dissolution chamber is 200 to 250 ml, which seems more biorelevant according to the latest studies than the standard 900 ml. In the second step, fraction absorbed ratios estimating food effect and bioequivalence were predicted based on the flux results with the help of the mechanistic model by Sugano. The same model was used to calculate fraction absorbed ratios from in vivo data published by medicines agencies. Finally, the calculated values of fraction absorbed ratios were compared. On this slide, the results of flux assays are presented. In case of all formulations, the donor media became very turbid right after the beginning of the experiment, disabling the in situ UV detection. For this reason, only the acceptor concentrations are presented in the figures. Let's see the acceptor result in fasted state first. For simulating the pH change when the formulation goes from the stomach to the small intestine in fasted state, media conversion from SGF to fast-safe was carried out. For the first 30 minutes, the donor compartment was filled with SGF and the acceptor chamber was not connected to the donor yet. After 30 minutes had passed, media in the dissolution vessel was converted to fasted state simulated intestinal fluid and the acceptor compartment was added to the device. Changing the pH triggered immediate precipitation in the donor compartment for Sporanox solution and further precipitation for Sporanox capsule. Suba-Itra capsule only disintegrated after media change was conducted because of the pH-resistant polymer matrix. Although the dissolution and precipitation kinetics of the two capsules were quite different, the appearance profile obtained is very similar. Itra was permeating circa 4 times faster from Sporanox solution in fastive than from any of the capsule forms. For simulating the conditions after food intake in the gastrointestinal tract, passive media was used in the donor compartment. No media change was carried out, and the acceptor chamber was assembled into the dissolution vessel from the beginning of the experiments, meaning flux experiment started from time 0. Although in case of the capsules, similar early flux results were obtained, the appearance profile of Itra from Sporanox capsule showed flux decrease after 2 hours, while no change in the case of Suba-Itra capsule. These profiles suggest that Itra dissolving from Suba-Itra capsule is in a quite stable solution with constant concentration of the drug in solution, while Itra dissolves and slowly precipitates from Sporanox capsule in a passive solution. Similarly to fasted state, the solution formulation outperformed the amorphous solid dispersions. First, let's see the food effect prediction based on flux results. Early fluxes were calculated from the linear portion of the appearance profiles presented in the previous slide. In case of Sporanox solution, the early flux is higher in the fasted state than in the fat state, while in the case of the capsule formulations, fat state seems more advantageous from the flux point of view. Fraction of those absorbed ratios was calculated for each formulation with the help of the equation written on the arrow. The FA ratio was also calculated from in vivo AUC data obtained in fasted and fat state. The comparison of in vitro and in vivo FA ratios can be seen in the figure appearing now. FA ratio 1 means no food effect in this context. If the value is less than 1, then there is a negative food effect, while if it's more than 1, a positive food effect can be expected. In case of Sporanox solution, the predicted FA ratios showed slightly negative food effect, which is in good agreement with the in vivo data, and also the recommendation to take the solution before meal. Sporanox capsule is advised to be taken after a meal, which seems reasonable considering the in vivo results. It can be seen from the right figure that in vitro-based FA calculation predict the in vivo behavior well. In case of the Suba-Itra capsule, the manufacturer claimed to have found a reduced food effect compared to Sporanox capsule, and according to the patient leaflet information, the drug can be taken regardless of meals. Though flux-based FA ratio seems to show slightly smaller positive food effect than in the case of Sporanox capsule, according to in vitro-based predictions, still some positive food effect can be anticipated.
After predicting the food effect from flux results, now let's focus on the comparison of different Hydroconazole formulations and predict whether they may be bioequivalent or not. First, we are going to compare the two capsule formulations. Fraction absorb ratio was calculated from in vitro flux results and from the in vivo AUC of the test and reference products as seen on this slide. According to the in vitro predictions in fasted and fed state, the FA ratio is around 1, which means that bioequivalence is expected. This prediction is in agreement with the in vivo data in case of the fed state, but as shown on the figure with the green bar, the in vivo data differ from the ideal 1 value in fasted state. Because of the significant difference obtained between the AUC values in the fasted state, the suba-ITRA capsule was found not bioequivalent to the Sporonox capsule. Repeated in vivo studies proved that suba-ITRA is therapeutically equivalent but not bioequivalent to the Sporonox capsule. While the composition of suba-ITRA amorphous solid dispersion, so the use of HPMCP instead of HPMC, might enable the rate of absorption to be very similar to the reference product, the half dose and the short gastric retention time in the fasted state might be responsible for the significantly reduced AUC value. These effects cannot be modeled by the in vitro method used in this study. This might explain the difference between the predicted and actual fractional absorb ratio in fasted state. Now I would like to present the comparison of the Sporonox capsule and the Sporonox solution. Sporonox solution was marketed 5 years after the capsule by Janssen Pharmaceutical. When launching the new formulation, it was compared to the capsule form. The Sporonox solution and capsule were compared in fasted state by brewers and co-workers. In the figure, it can be seen that the flux-based fractional absorb ratio gives a fair estimate to the in vivo results. Both are in agreement that from Sporonox solution, at least 3 times higher amount of Hydroconazole absorbs than from the capsule formulation. When the solution formulation was launched, it was already known that the solution was recommended to be taken before a meal, and the solid formulation should be taken after a meal. Therefore, the Sporonox solution in the fasted state was compared to the Sporonox capsule in the fed state. Both in vitro fractional absorb predictions show superior bioavailability of Hydroconazole from the solution, which agreed well with the in vivo results. The in vivo fractional absorb ratio of Hydroconazole from Sporonox solution was found to be circa 2.5 times higher than from the capsule formulation. Since the bioavailability of Hydroconazole was greater from Sporonox solution, therefore the solution and the capsule were not bioequivalent products according to FDA. In conclusion, we can say that flux-based predictions were found quite efficient in the prediction of the food effect and non-equivalence of different Hydroconazole-containing formulations.
In our next case study, we tested if even small differences between brand and generic formulations can be seen with flux studies. The macroflux device was used to run the study, which is a receiver chamber with permeation membrane, overhead stirrer and fiber optic UV probe inserted in the 900 ml vessel of the USP2 apparatus. Talmisartan was selected as a poorly water-soluble model compound, and the brand name formulation Micardis was tested against several generic formulations. In this presentation, I'm only going to show the results for 2 generic products, Ratioform matching the composition of the brand, and Actavis, which has a slightly different composition. You can find further examples and details in the cited article. First, let's see the dissolution and appearance profiles of Talmisartan from the brand tablet Micardis and the generic Ratioform. They both contain sorbitol as a tablet filler, and other components of the tablet are also qualitatively identical. The dissolution profile for the formulation showed slow release kinetics in SGF, but after the media change to fast-sieve, the API concentration rapidly reached its maximum. The appearance profiles show that in the first 30 minutes of the experiments, no detectable amount of API could cross the membrane, since the double protonated form of the API was not able to permeate through the lipophilic barrier. After media conversion, the transport of the molecules became much faster because of the presence of the uncharted form in solution. In order to predict bioequivalence or non-equivalence from the in vitro flux results, the flux ratios and 90% confidence intervals of generic formulations were calculated from the log-transformed fluxes. This is the same way calculations are done during the in vivo human bioequivalence studies. In the case of these studies, the acceptance criteria for bioequivalence is that the 90% confidence interval must fall within the 80 to 125% range of the reference. In this case, the flux ratio showed a perfect match, and the confidence interval fell well within the acceptance range. So based on the in vitro results, Ratiopharm is expected to meet the bioequivalence criteria. From the table at the bottom, it can be seen that the in vitro and in vivo results were in agreement, showing that the Ratiopharm formulation was found to be bioequivalent to the brand formulation. Now let's look at a generic formulation with slightly different composition than the brand name. The dissolution and appearance profile of telmisartan from brand Micardis and mannitol-containing generic formulation Actavis are shown in the figure. The dissolution profile from the generic formulation was found to be quite different from the brand, showing much slower tablet disintegration and delayed dissolution after media change. Though the generic form showed slower release kinetics than the Micardis, they reached the same maximum concentration after 2 hours. If we take a look at the appearance profiles, it can be seen that the membrane transport was faster in the case of Micardis than in the case of Actavis tablets. The composition of the Actavis tablets only differed in the type of the filler. Actavis contains mannitol instead of sorbitol. In the case of the mannitol-containing formulation, the in vitro flux ratios should decrease compared to the Micardis tablet, and the lower confidence interval fell out of the acceptance range, as can be seen in the table. The in vivo Cmax values showed a similar pattern since the Cmax ratio was found to be in the acceptance range, but the lower limit of the confidence interval was not. The public assessment report of the generic formulation states that the formulation was on the borderline of the acceptance range, and the confidence interval most probably did not fall within the range of 80 to 125% because of the high standard deviation in the in vivo results. This explanation has been accepted by the regulatory agency, and the Actavis tablet was also accepted as a bioequivalent product. In the previous slide, we could see that a generic formulation with a different tablet filler produced significantly reduced flux values and almost failed the bioequivalence test. In order to investigate whether it was because of the effect of excipients on the absorption of telmisartan, 80 mg of mannitol was pre-dissolved in the donor media while measuring the flux from the brand tablets. Mannitol as a pre-dissolved additive did not affect this solution, but did have an effect on the flux, therefore also on the permeability, resulting in slower membrane transport than without the excipient. Using this technique, the importance of excipient selection could be demonstrated. In conclusion, we can say that flux assays could be great in vivo predictive tools before bioequivalence studies are conducted, because they can help to understand the effect of additives on this solution and membrane transport by identifying the excipient responsible for flux increase or decrease. All in all, it can provide useful feedback for formulation developers.
In the final portion of the presentation, we would like to show an example of how the ADDF concept can work in a formulation development process. In this demo project, we have applied an extremely simplified approach to formulation development and have not considered any usual formulation criteria like manufacturability or stability, and have only focused on controlling the flux of the API in each step. Telmisartan has been selected as model compound primarily due to the previously presented Micardis example, where the large volume flux provided a reasonable prediction of the in vivo studies. This encouraged us to consider the final flux assay as representative of the in vivo behavior. As the first step, we have carried out an excipient screening on the Pampa platform. The excipients of the available temozoltan formulations and commonly used standard excipients were involved for the first API-excipient interaction investigations. Due to the experimental differences in the applied doses, the flux data were considered misleading and were normalized by the donor concentration that resulted in the apparent permeabilities on the y-axis of the graphs. The left graph shows the effect of surfactants, where significant permeability reduction compared to the excipient-free case has been found, suggesting that those are not ideal excipients for this formulation project. The polymers in the middle graph show a slight, non-significant increasing effect compared to the reference. As shown on the right side graph, mixed effects were experienced with fillers, where mannitol provided a lower permeability than the others. For the later stages of the demo development, tween-80, both PVP versions, HPMC-AS, sorbitol and mannitol were used in the formulation samples. Temozoltan is known to be exceptionally stable in amorphous form, therefore the strategy of preparing an amorphous solid dispersion-based formulation has been selected. Both PVP and HPMC-AS were selected based on the excipient interaction study, and amorphous solid dispersions were prepared using electrospinning technology. The scanning electron microscopy images of the electrospun samples confirmed the formation of thin fiber structure in case of PVP-containing formulations, while no fiber formation, only electrospraying occurred when using HPMC-AS. Tween-80 was added to optimize surface tension of the polymer solution, which not only helped the formation of fibers, but also significantly increased the productivity of the process. The amorphization has been confirmed by X-ray powder diffraction and DSC measurements. These samples have been found to be suitable for small volume flux testing. The small volume flux testing was performed using microflux apparatus. The media conversion assay protocol was carried out as described previously. As shown on the left graph, all 4 formulations dissolved rapidly and were completely dissolved in about 30 minutes. The right side graph demonstrates that the permeations only started after the media change, and formulations containing tween-80 show significantly poorer flux values than formulations without the surfactant. The permeability graph also shows the same significant effect of tween-80, while only negligible difference can be seen between formulations prepared with different polymers. In spite of the high productivity of the tween-80 containing formulations, the flux assay results prompted us to select the 2 electrospun formulations prepared without surfactants for the next step, final dosage form preparation. As only a small difference has been seen between the PVP and HPMC-AS based solid dispersions, both versions have been moved to the next stage of final dosage form preparation. In the excipient screening, sorbitol was found to be one of the most advantageous filler, while mannitol was the worst. Tablets were prepared with these 2 fillers in order to confirm the expected difference in the final dosage form as well. 4 different tablet versions were made as per the table on the left, varying the polymer and the filler in the tablets. These tablet samples were studied in the large volume flux study. The macroflux system coupled with in situ fiber optic concentration monitoring has been used in this final investigation. A biorelevant media conversion protocol, as explained before, was used in the dissolution vessel. The donor data on the left side shows a delay in disintegration and dissolution for the HPMC-AS mannitol tablet, but it also reached 100% dissolved after about 60 to 70 minutes. The permeation to the acceptor started after the media conversion, and the flux was constant in the study time frame. The permeability graph on the right gives a quantitative comparison of the formulations. In case of PVP-based tablets, the use of mannitol or sorbitol did not have a significant impact on the behavior, unlike HPMC-AS-based tablets, where the mannitol version provided a lower permeability. This formulation, so the HPMC-AS mannitol, was the lowest performing in the whole study. The use of PVP versus HPMC-AS has not resulted in a significant difference in case of the sorbitol tablet versions. As a result of the formulation project, 2 out of the 4 developed tablets performed very similar to the brand formulation of temozoltan, showed with the red bar, so both sorbitol-containing tablets can be expected to perform equally well in vivo human studies. Also, the early indication of sorbitol's advantage over mannitol has been confirmed in the final dosage form as well. It is interesting that we have observed a similar pattern in this study as was found in the brand and generic formulation comparison of telmisartan, where the flux data and also the in vivo data showed a better performance for the sorbitol-containing tablets over the mannitol-containing ones. The absorption-driven drug formulation concept has been created to understand the effect of formulation-related decisions on flux of the API and to help the formulation scientists to predict the in vivo human absorption more precisely. The presented case studies aim to provide a broad view on how various flux assays can assist with excipient or formulation strategy decisions, and the demo project provided a simple example of how the concept can be implemented in the complete formulation development process. This project could not have happened without colleagues and friends at the two universities and at Pion. We are grateful for their support.